So thank you. Let me first of all thank the organisers for the invitation to participate in this meeting. It's absolutely been fascinating over the past 2 days. It's not an area that I've really delved too much in, but obviously has a major impact on maternal and fetal well-being during pregnancy. So I've been tasked with talking about the role of the placenta in delivering nutrients and in developmental programming. So I want to start out here showing you a picture of the triad of mother, placenta and fetus. And obviously it's the mother who takes in all the nutrients for pregnancy, so she's exposed to different levels and different types of nutrients. But she provides this nurturing intrauterine environment that contains the placenta and fetus. And you see there's a three-way interaction between each component. The mother supplies the nutrients, the placenta takes them up and transfers them to the fetus, removes waste products, but the placenta also communicates by various means with the mother and the fetus. So if we have three components, we've got to say who's in charge. So I believe firmly that the placenta is the director of pregnancy. It's in charge. We could have a debate about it afterwards if you like, but that's my viewpoint on it. And you'll see here at the bottom we have a fetus, and it's coloured pink. It's coloured pink on purpose. It's a girl. It's good to be a girl in utero. Conversely, it's not so good to be a boy in utero, and I'm going to come back repeatedly to this idea of sexual dimorphism in the fetal responses, but also sexual dimorphism in placental function as well. Obviously, you have a female fetus, you have a female placenta, and vice versa, and it took us a long time to begin to appreciate that, and it's only really over the last 5 years or so that people have started talking about sexual dimorphism in the placenta. And I was also asked to talk about what has changed in the past 30 years. So in the top right here shows you what's changed in the last 30 years. That's the increase in obesity and diabetes in the population, which has really impacted outcomes in pregnancy. And if you think about it in evolutionary terms, probably throughout the entirety, virtually for this last little blip of our evolution, we've faced nutritional challenges. And probably we've had deficits in nutrition, and the placenta and the fetus have learnt how to cope and to adapt with these nutritional deficits, so it has mechanisms in place for that. But now we've completely reversed the picture, and we're faced with this excess of nutrients in the settings of obesity and diabetes, and what does that then do to placental function, fetal development, growth, and programming? So I was asked to talk about developmental or fetal programming. We've heard a fair bit about it so far. Originally called the Barker hypothesis after Sir David Barker, who did a lot of description in this field, now commonly called developmental origins of health and disease. Simply stated, your life in utero determines your risk of development of disease in adult life. So if you're exposed to some adverse condition, could be good or could be bad, potentially, in utero, it will program fetal systems and you will develop diseases in later life, particularly cardiovascular disease, diabetes, metabolic syndrome, etc., and a lot more emphasis now down at the bottom on neurobehavioral outcomes as well. And again, as we've heard today, there's a sexual dimorphism in effect here. So differential effects to the responses to the same insult, whether you've got a male or a female fetus, and often in a lot of cases, the males are more severely affected than the females. So what are the mechanisms that really give this developmental programming? So obviously differences in type, level of nutrition are important, and we're going to talk a bit about that. And there are physical factors that affect fetal development. So for example, my colleague sitting over there, Dr. Thornburg, knows that increased vascular resistance in the fetal placental circulation in a growth-restricted placenta alters the strain on the fetal heart during development, and so you alter fetal heart development and you program potentially for cardiovascular disease. The mother is exposed to environmental contaminants, pesticides, etc., which again can be transferred from the placenta to the fetus and cause developmental programming. And most of these factors seem to act ultimately by epigenetic mechanisms. By epigenetics, we know that there's DNA methylation that we've talked about, which alters gene expression, and histone modifications, which affect chromatin structure and again alter gene transcription. So probably most, if not all, act by epigenetic mechanisms. So let me briefly review the roles of the placenta. So the placenta is the immune barrier between mother and fetus. It prevents rejection of the semi-allogeneic fetus. obviously. It's the site of exchange of oxygen and nutrients. So oxygen diffuses across to the fetus, then carbon dioxide may be removed by diffusion back across the placenta. But the placenta is a veritable factory for steroid and peptide hormone production. It produces grams and grams of peptide steroid hormones each day, along with other metabolites. These are released to the maternal circulation where they can affect maternal metabolism, increase availability of substrates, glucose, fatty acids, amino acids, for transport to the fetus. Also, the plane of nutrition that the mother is under, the influence of obesity and diabetes, are also going to alter the amounts of nutrients in the maternal circulation. These nutrients are then taken up by the placenta by transporters on the placental surface. They could be transported straight across to the fetus to affect fetal growth and development. However, a substantial proportion are also metabolized by the placenta to generate ATP, which drives steroid synthesis, drives all the transporters that are there. In addition, some of these metabolites might actually also be transferred to the fetus. So, for example, there's a lot of interest now in serotonin, which is produced by the placenta, having a role in fetal brain development. So again, placenta influencing fetal development. So I want to emphasize that the placenta is not just a conduit. In the human, the placenta consumes 50% of all the oxygen which is supplied to the uterus, so the other 50% is probably available for the fetus, and it consumes 30 to 50% of the glucose supplied to the uterus. So we heard yesterday that the fetal brain needs 32 grams of glucose per day, so it's likely that the placenta at least requires 32 grams or so of glucose per day. It's using that. The metabolic activity of the placenta is 4 to 6 times higher per unit weight than the fetus. So the fetus is about 6 times larger at term, but the placenta has a much higher metabolic activity, hence it's consuming about half of the fuels and oxygen. And of that oxygen that's consumed by the placenta, about a third is used for de novo generation of peptides, about 1/3 to maintain the cation gradient across the membrane for transport. So it's not simply a conduit, it metabolizes a lot of the nutrients, and it's referred to now as a selfish organ. It regulates nutrient composition and supply from the mother to the fetus, and maybe the fetus gets what the placenta doesn't consume. And if you think of this again in evolutionary terms, you cannot have a fetus without a placenta. You can have a placenta without a fetus. And in terms of actually supporting the fetus, adapting throughout gestation to respond to adverse conditions, does the placenta adapt, alter metabolism regulation to try and maintain fetal growth? And it might be smaller, but it tries to maintain that to prolong the life of the fetus. So what about mechanisms of transport? There are several. One is flow-mediated diffusion. Obviously, that's how oxygen ions, small ions, get transported across the placenta. The rate-limiting factor is blood flow on both sides of the placenta. The general rule of thumb is that compounds with a molecular weight of less than 1,000 can actually simply diffuse across the placenta, but there are obviously exceptions to that. Things that need to be transported across a concentration gradient or against a concentration gradient obviously don't diffuse. They have to be actively transported. So folate, for example, would be a good idea of that. There's a lot of discussion whether fatty acids freely diffuse as they're lipophilic, but I think most of the evidence suggests that specifically transported by fatty acid transporters. So active transport occurs when you need to transport against concentration gradient. So amino acids are a good example of that. Facilitated transport down the diffusion gradient, so glucose and fatty acids, which require transporters, but they're going with the concentration gradient. And then for large molecules, proteins, things like IgG, insulin, they're taken up by endocytosis, exocytosis. So then how does the placenta mediate the effect of changes in nutrition on health and disease? So as I've tried to explain, it can regulate the type and amount of nutrients that are transferred. It definitely regulates maternal supply. So placental lactogen, growth hormone, etc., are released into the maternal circulation, alter maternal metabolism to free up substrates for transport. So it regulates the supply. It seems to regulate fetal growth and development, so that's the demand that the fetus is making on the placenta. It obviously can alter the expression of types and quantities of these various transporters, so alter the amount of nutrients. It may buffer and store certain nutrients again, so influence transport. And we know if you alter placental metabolism that consumes or produces different nutrients and fuels, that will alter or limit transfer to the fetus. So again, it doesn't simply push everything to the fetus. The placental metabolism itself regulates what gets to the fetus. And it's probable, as I say, that from evolutionary pressure, there are adaptive responses to altered supply. And does the placenta increase or decrease the transport of nutrients and alter production, metabolism, or storage as part of these adaptive mechanisms to ensure survival of the fetus? And obviously, it can do so by altering the regulatory signals to the mother and the fetus, and obviously then the epigenetic responses as well are probably part of this adaptation. So I want to come back again to sexual dimorphism. So as we've heard, we know there's sexual dimorphism in fetal outcomes, and it's apparent that we've evolved different evolutionary strategies for males and females, and there's very good evidence from the animal literature that we can alter the ratio of male and female fetuses by altering nutrient composition and supply to the placenta. So for example, the red deer, the mouse, if you alter their environment, their nutritional plane, restrict certain nutrients, they can alter, usually reduce the number of male fetuses to maintain the number of female fetuses, obviously as an evolutionary response to trying for continuation of the species. Okay, so they seem to change males and females in response to nutritional stress. You can actually feed mice omega-3 polyunsaturated fatty acids, and you can increase the ratio of male pups as well. In humans, male fetuses appear to want to keep growing throughout gestation. They're usually larger at birth, but they also suffer more adverse outcomes as a consequence. So by virtue of wanting to grow large, they're susceptible then to many stresses. So we know there's more preterm births, placenta previas, preeclampsias, their lung development lags behind. They're more macrosomic fetuses, they're late stillbirths in the fetus if it's a male. And being a male fetus, male placenta can also affect maternal beta cell function, which is poorer if you have a male fetus, and increases the risk of gestational diabetes. So it translates not only at the placental level, the fetal level, but back towards the mother as well. In contrast, females adapt their growth rate to optimize survival in a poor environment, because obviously we need the females for reproduction, ultimately. And we know that there are differences in fetal programming, for example, of the metabolic syndrome based on the sex of the fetus. But the sexual dimorphism also happens at the level of the placenta as well. And there are several papers now published that show differences in gene expression between a male and a female placenta, both in the first trimester and at term. And this has more lately been linked to an escape from X chromosome inactivation that occurs. We know there are differences in inflammatory, hypoxic, apoptotic, autophagic responses between male and female placentas, differences in expression of antioxidant defense enzymes, which I'll show you some data, differences in fatty acid transporters and fatty acid oxidation. I'm going to show that data as well. Differences in response to maternal adiposity and inflammation, differences in microRNA, steroid synthesis, etc. And they're all linked to difference in outcomes of male versus female fetuses. I just want to show you as well that the placental growth and development is not static, obviously, across gestation. A first trimester or a 6-week placenta is not a miniature version of a term placenta. It's a constant state of growth and differentiation across gestation. So in fact, at 6 weeks, the placenta is bigger than the fetus, whereas at term, obviously, the fetus is about 6 times bigger than the placenta, and the ratios are obviously reserved. And again, the structure function changes across gestation. So, for example, at term, the proportion of the placenta, the villus placenta that's occupied by the vasculature is about 28% versus 2.7%. So it's obviously very highly vascularized at term. The trophoblast surface area is 0.1 or so of a square meter, which is about the size of both my feet on the ground. So that's at 6 weeks, whereas at term, the surface area of the placenta is 12.5 square meters. To show you what that is, that's the size of a parking spot. Okay, is the size of the surface area of the placenta. That is the exchange area, which is the size of a parking spot. It's full of transporters, et cetera, et cetera. So massive area for uptake of nutrients. The trophoblast thickness and maternal fetal diffusion distance also decrease as you go towards term. So the net result is a placenta with vastly increased efficiency for transport of nutrients as you progress throughout gestation. So there are obviously many critical periods across placental development. So the timeline is for human placental development, and you see different things occurring at different times. So if you were to put in an insult at any specific time point in placental development, you might have a specific effect that alters placental structure function. If you put the same insult in at different times, then again, you might have completely different effects. And this is very nicely shown again in animal models, particularly the mouse, where if you affect the level of nutrition, the type of nutrition, the duration of change in nutrition, etc., give the mouse high fat or high protein, or change it or alter the oxygen level that the mother's inhaling, you can alter placental structure across gestation. So it looks like the placenta can adapt to these insults, presumably as an adaptive response to try and keep the fetus alive. It's obviously much more difficult to study this in the human placenta, but there is evidence that it happens. So at the bottom, my colleague Thomas Janssen showed quite a few years ago that if you took a woman who had insulin-dependent diabetes mellitus, so she was hyperglycemic at the beginning of gestation, she ended up with a LGA, large for gestational age baby, and he saw increased GLUT1, the main glucose transporter in the basement membrane of the placenta, and increased system A amino acid transporter. Whereas if he looked at placentas from women who had gestational diabetes, i.e. their hyperglycemia started later in gestation, they could get an LGA baby, but there was no change in GLUT1 in the basement membrane. So the same insult, i.e. hyperglycemia, different time points in gestation, you get different responses. Okay. So what about the influence of nutrition or metabolic environment on epigenetic modifications? So the epigenome responds to changes in nutrients, including methyl donors, folate supplementation, fat, glucose, and caloric restriction. We know that now. And one of the earliest illustrations that this was occurring was when the offspring of women who were exposed to the Dutch hunger women were examined, and differences in DNA methylation were found in women, or the offspring of women who were or were not exposed to the Dutch hunger winter, and whether they were pregnant to the first trimester or pregnant to the third trimester at the time of the hunger winter as well, differences in DNA methylation. And we know that variations in DNA methylation are associated with many aspects of diabetes mellitus, for example, and metabolic inflammatory milieu of obesity. And I'm going to go on and show you some data on that. So what is the linkage between nutrition and epigenetic changes? So shown on the left, many of the challenges that can be seen in animal models or occur in the human scenario: caloric restriction, obesity, diabetes, increases in glucose, fatty acids, bioactive food compounds, folate, etc. And what they do, obviously, is they alter one-carbon metabolism, as we've heard described yesterday and today. They alter metabolism through the TCA cycle in the mitochondria. That affects the level, for example, of substrates like alpha-ketoglutarate, which are generated in the TCA cycle, which is a cofactor in DNA methylation and hydroxymethylation reactions. So the net result being we see alterations in DNA methylation and hydroxymethylation, alterations in histones, which affect chromatin structure, then affecting placental gene expression. That leads to alterations in placental function and ultimately leads forward to developmental programming. And we can see these occurring in the placenta, but the placenta is a fetal tissue, so it's also likely that a lot of these epigenetic changes occurring in the placenta also occur in the fetal tissues. What we're not certain of is whether they're identical in placenta and fetus. Probably not all of them are, but probably a lot are similar. So we see epigenetic changes throughout the fetal placental unit in response to these inputs. And we talked a lot about micronutrients yesterday, and obviously a lot of these micronutrients can affect epigenetic modifications. So shown here is the one-carbon cycle. So we've got folate, vitamin B6, choline that we heard about yesterday, betaine, vitamin B12, obviously components of the one-carbon cycle. So if we alter the amount, we're going to affect the one-carbon cycle. But then there are a whole host of other micronutrients now we know that affect epigenetic modifications. For example, butyrate and curcumin are shown to be histone deacetylase inhibitors. So they affect the histone conformation and the differences between the unmodified and methylated chromatin, etc. So again, many nutrients that can impact on epigenetic modifications. And again, what we don't know is how much is enough, is too much a bad thing, etc., or too little a bad thing. And we actually investigated now, about 4 years ago, the effect of obesity on placental DNA methylation. We published this paper in 2017, and actually what we did was just used a methylation array to look at the number of sites that were methylated in the placenta from lean women and the placentas from obese women. And what we saw was that across all tiled regions on the array, there was about a 15% increase in methylation of DNA. There's also a corresponding decrease in hydroxymethylation. We looked at different regions of genes, and we looked at 100 base pairs upstream from and downstream of transcription start sites. You can see about a 15% increase in methylation. If you look further out, for example, CPG island shelves, these big flanking regions, again, differences in methylation. And what we saw from this array, there were, I think, 262 genes where we saw reciprocal increases in methylation, decreases in hydroxymethylation. Quite a few of them were on chromosome 17 and chromosome 19. In particular, on chromosome 17, we saw human growth hormone, human placental lactogen, with alterations in methylation, and then pregnancy-specific glycoproteins on chromosome 19. And again, often these changes in methylation translated through to differences in gene expression. So this, I think, is a good illustration that obesity can alter placental DNA methylation. So what about oxidative stress? In pregnancy, we heard a lot about it yesterday. Obviously, antioxidants protect cells from oxidative stress that causes cellular damage. Normal pregnancy is a state of increased oxidative stress. We see further increases in pathologic pregnancies, like preeclampsia, diabetes, etc. The placenta is a major source of oxidative stress due to its high metabolic activity, and the mitochondria in the cells are a major source of reactive oxygen species production. And again, what we showed was that the inflammatory conditions of obesity and gestational diabetes heighten the oxidative stress in the placenta, and they deplete antioxidant defenses, often in a sexually dimorphic manner. So in this paper here we published 2017, what we showed was that in lean women, placentas of male fetuses had the highest antioxidant defenses. But if you looked at obese women, then the male placentas then had the lowest antioxidant defenses, which coincided with the highest levels of oxidative stress. So oxidative stress went up, and you got consumption of those antioxidant defenses in the male placenta to try and counteract that. So it's the male that seemed to be suffering the most versus the female in that condition. We talked yesterday about sources of antioxidants, and obviously the nutritional and supplemental antioxidants, but there are many other types of antioxidants within a cell, intracellular reducing elements, the enzymes like superoxide dismutase, glutathione peroxidase, etc., then extracellular antioxidants. So reactive oxygen species are produced in various locations within a cell. Their production is probably context-specific, and within those specific regions, there are specific mechanisms set up to actually scavenge those reactive oxygen species. So, for example, in the mitochondria, which produces a large amount of superoxide, the mitochondrial manganese superoxide dismutase and glutathione peroxidase are right there ready to mop up those reactive oxygen species. So perhaps it's not surprising with our failure of things like vitamin C, vitamin E to actually be able to effectively reduce oxidative stress if they're actually not reaching the site of reactive oxygen species production. So briefly, selenium, we've heard about it again. It's important, it's a trace element, but it's in the active site of glutathione peroxidase and certain forms of thioredoxin reductase. It's also a component of some unusual amino acids, and selenium deficiency in soils is associated with increased incidence of preeclampsia and dietary supplementation recommended and being studied in certain areas. So finally, I just want to talk a bit more about obesity and GDM. Obviously, these are the metabolic conditions that we see with obesity and GDM. Again, programming of the offspring for disease in later life, sexually dimorphic responses. And we actually have shown that with increasing maternal adiposity, we see decreased mitochondrial respiration. And we think that might be related to the increased incidence of stillbirth we see with obesity, that eventually the placenta falls off the cliff, its mitochondrial respiration is reduced that much. Gestational diabetes, it seems to be even worse, but the placenta again has got an adaptive response. So with gestational diabetes, where respiration is down, it compensates by a bigger placenta, usually a gestational diabetic, and increased glycolysis to try and generate energy as well. So again, does that prevent total collapse in gestational diabetes? We look at the substrates that the placenta has available, looking at mitochondrial respiration here. There are three main substrates: glucose, fatty acids, glutamine. Different cell types prefer different substrates as their main source. So, for example, the brain likes glucose, the heart likes fatty acids, tumor cells like glutamine. And interesting, glutamine is by far the most abundant amino acid in the maternal circulation in pregnancy. It's about 10-fold higher than any other amino acid, about 900 micromolar. So what we did was to look at the contribution of each of these substrates to placental trophoblast metabolism of the mitochondria. And you can do that just by employing specific inhibitors. And often cells have the ability to switch between use of different substrates. So if one becomes limiting, it can switch to use of another one. That's called metabolic flexibility. Okay? So what we found was in trophoblast of lean women, there was no difference in dependency on each of these three substrates for baseline respiration in a male or female placenta. Each substrate contributed about a third of oxygen consumption in a lean placenta, no difference between male and female. But with hyperglycemia and hyperlipidemia of obesity and GDM, we found increased dependency for use of glucose and fatty acids, but only in a male placenta. So you give it more glucose, give it more fatty acids, it uses it, but only if it's male. And the accompanying that was a significantly decreased flexibility. So they like using the glucose and fatty acids, but they then can't switch easily to using glutamine or whatever. And again, is this related to increased risk of adverse outcome? And the effect's not due to obesity alone, but we think is due to this worsening hyperglycemia and hyperlipidemia that we see. And again, ultimately, does this mean that changes in placental metabolism affect what ultimately reaches the fetus? So I just want to briefly, the last few slides, I apologize, just talk about fatty acids and brain growth. So we know that DHA and arachidonic acid are essential brain-specific fatty acids. Brain growth increases dramatically in the third trimester and postpartum. And interestingly, I saw this paper, Ogundipi et al., from some colleagues in London a couple of years ago, and they showed that brain-specific fatty acid supplementation in pregnancy, where they looked at brain size after supplementation determined by MRI, and found that only males born to women who'd been supplemented with these brain-specific fatty acids had bigger brains. So again, a sexual dimorphism, it looked like the male fetus was able to pull in or take in more of these brain-specific fatty acids. That fits, I think, with its need to keep growing in gestation. If we talk about fatty acid metabolism, I'm going to talk a little bit now about fatty acid beta oxidation and generation of ATP in the placenta. Suffice it to say, fatty acid oxidation is undertaken by a family of enzymes which show specificity for either medium and short chain fatty acids or long chain fatty acids. So ACAD-M is the acyl-CoA for medium chain versus ACAD-L, etc. And what we showed, we looked at expression of these enzymes in male versus female placentas, the influence of obesity. We see a sexual dimorphism in expression of the enzymes in placental tissue. And with obesity in the male, there's increased expression of enzymes that prefer the short-chain fatty acids, whereas the female has more enzymes that prefer the long-chain fatty acids. And is this a difference in response to the fatty acids reaching the placenta, or is it an attempt by the male placenta to produce energy from short-chain fatty acids, which are usually more easily available, and then alters the fatty acid composition that gets transported to the fetus? And for example, I showed you the fetus likes DHA. So the final couple of slides. So this is a lipidomic analysis now of fatty acid composition of various lipid classes in the placenta from lean, obese, and gestational diabetic women. At the top, yellow is female, blue is male, and the action is in the bottom right-hand corner there, where you see the green, all the green, and this is decreased expression or decreased amounts of fatty acids, triglycerides, which contain fatty acids with 22,6, which is DHA. So we've got less DHA in triglyceride species in the male placenta of the A2GDM. And I actually went back last night and looked at phosphatidylcholine, which had been mentioned yesterday, and indeed, phosphatidylcholine, there's less DHA in the males as well. Okay. So just final, so to put that together, my apologies, fatty acid oxidation enzymes increasing in the enzymes that like the short-chain fatty acids and medium-chain fatty acids in the male. There's more medium-chain fatty acids in the male and less of that DHA. And that parallels quite nicely some data that was published in 2018 by Kathleen O'Neill, where she looked at fatty acids in amniotic fluid. And again, there were more medium-chain fatty acids in amniotic fluid of diabetic women with a male fetus versus more long-chain with a female fetus. So the take-home message is the placenta directs maternal metabolism to promote fetal growth and development. It responds to alterations in nutrient supply to ensure fetal survival. These responses may developmentally program the fetus via epigenetic mechanisms. At delivery, we can use the placenta as a diary of fetal exposures, and a lot of people actually do that. There's obviously sexual dimorphism of effect and function. And what I'm saying now, I think, is that if we're going to look at interventional or supplementation studies, we need to consider sexual dimorphism, and we need to look at the long-term effect. Are we interfering with some adaptive effect and potentially do something harmful, or are we actually trying to interfere with a primary dysfunction of the placenta in which we were trying to improve things? So thank you very much.